Okay, thank you very much. I'm Miguel García Hidalgo, and I'm from the Cambium Research Group that belongs to the iuFOR Institute in Soria. And I'm happy to be here, and above all, I would like to thank the previous talk, because we can learn a lot of the technology today. I'm working somehow in something similar to Chen, but in other perspective. Chen was using CT images, which are very useful depending on density, wood density, for example. But we are working in our group trying to mix two really good perspectives to know how is the evolution of trees and forests. One way is remote sensing, and the other one is dendrochronology, because mixing both fields, we can get reliable data from the actual and day-by-day perspective, and from the past perspective. That is the main limit of remote sensing, that we don't have many data from time ago. So we work with, well, it's not saying very well, but I try to talk about what it should be seen on these images. We're working on trying to discover how trees and how forests will respond to the actual and the current climate scenario. So we are getting some data from trees by using tree cores. That is quite a bit destructive for trees, but they can survive and they can improve their growth and continue with their life. And we get the essential data from cell to cell in the growth. So we are trying to discover how trees respond cell by cell to the environment, just with different kinds of cells, trichates and conifers, for example. But in not only ringweed, that is the main approach to know about the tree growth, but trying to know the vessel size, the ray parenchyma, or other kind of standards and measurements that we can get from this kind of samples. We make microslides from these three cores to discover the cell distribution and the cell patterns for each ring. We have to take into account that the meaningful information of dendrochronology is if we can tell the difference between rings. Because if we know that some cells belong to one ring, we can correlate this information with the climate, because we know how was the precipitation of these years. For example, we correlate with mean per month precipitation or temperature, so on. And I would like to show some results of some of our studies. Everything started when we worked with these images, and we tried to get all the cells that are in the image. That is a really hard-working issue and really time-consuming, because, by the way, we usually do it by common standards of image analysis, or with thresholds and only with pixel color value, not taking into account their position on the image or other things that can be useful for this. And it was really time-consuming, expensive, and sometimes it is not accurate at all. So we wanted to develop some new techniques to improve this data, and we developed a first model using deep learning algorithms based on unit R deployment methods, and we get the results of cell-by-cell segmentation. But that is not useful if you don't have the ring limit. The ring limit must be done by the technician, by specialized people trained to do it, because different species have different patterns and different ways to detect the rings. So, for this second approach to detect the rings automatically, we get 214 slides with, which were tagged by 2 different users to improve the variability of the data, and we get almost 6,000 rings delimited. So we bought these rings, and we tried unit R-based model using Python and Python. And after that, we trained with the 60% of the data. We evaluate the results with 20% of the data, and we tested the best models with other 20% of data. Well, we used this kind of network, that is unit R with a VIT method in order to transform better the images. There was RGB images in GPEJ format, so it's not good at all because you have a loss of the images, but it can work really well with this kind of images. So the results were, as you can see, over. The essential or the current way to evaluate if a ring is detected or not in the field, in the technology, is if the ring is detected, then the ring is right. And okay, that was good. But if it is not detected, well, it is wrong, the ring. If we evaluate this way to get data from this algorithm, we reached almost 100% of results. So almost all the limits were detected. But if we used the standard ways to evaluate deep learning models, the results were almost 0. And why? That was the worst question that we have to answer in this paper. Maybe the ways to evaluate these methods, these deep learning methods of segmentation, are not accurate for this field. We have to take into account that mainly the whole images are not rings. So we are limiting just 1% of the image, of the pixels of the image. So we have to evaluate this kind of segmentation. If you have a border in the same ring, but just 1 pixel away, the result is 0. But it is not relevant for the quantitative wood anatomy study. So it is not a good way to evaluate. We decided to evaluate it by an expert. And then the results were that, taking into account that the cells were integrated to one ring or to another, that it was the best way to evaluate if the ring was properly detected and bordered. Then we reached that the model was similar or better than the expert segmentation that was for the training method of the main segmentation method. Yes, in the percentage of rings number, in the main ring center difference in pixels, but they were really, really low, 12 pixels of difference, but it wasn't a difference into the cell mismatch of the rings. So we get that the way to evaluate this kind of algorithms must be adapted to the field that we are applying them. So that was all. Thank you very much.